We got to beginning the discussion of positive feedback, and we're talking about the way or the ways in which that positive feedback differs from negative feedback. So let's just sort of step back and review what we just said about negative feedback. Negative feedback is really a change in the opposite direction with regard to our different regulated variables. So our examples we gave were body temperature, blood pH, body composition, and so forth. Glucose was another example that we discussed. So when we have a change that deviates these variables away from our set point, which is where we'd like for them to be under homeostasis, that change, in order to correct it, needs to go in the opposite direction. So the feedback loop is a way for us to return that variable back to its set point, and negative feedback is the mechanism for doing so. Positive feedback, on the other end, is usually a part of a negative feedback cycle. However, positive feedback is where we sort of augment or amplify that change. So this is going to stimulate the process or sort of potentiate that change, moving us away from set point further, but that is for a short or fixed period of time, and that is for a desired result, and that will ultimately end with a negative feedback loop. So a positive feedback loop only temporarily contributes to a negative feedback loop. Let's look at some of the examples in our body. Now, as we said earlier, it's much more rare than negative feedback. So there are very few examples of positive feedback that we can identify. One is when we injure or damage a blood vessel. Let's say you get a paper cut, for example. Now, you're going to see some bleeding initially, and the process is going to allow for the formation of a blood clot in order to stop the flow of blood. So the way that we see that process is for there to be a little bit more bleeding temporarily, which can bring clotting factors to the site of injury. It can bring certain proteins to the site of injury. And while that is potentiating the bleeding, which is a change that is moving us further away from set point temporarily, this is a part of the desired result. In order for us to clot that injury, we need to deposit some of these clotting factors, etc., at the site. And then when the site of injury is healed, it ultimately ends with a negative feedback, which is stopping the bleeding. So this is an example of how clotting, for example, involves negative feedback. But again, the principles of negative feedback is a change away from set point for a fixed amount of time for a desired result that ultimately ends with a negative feedback. Let's look at another example at the bottom here. This is the strength of uterine contractions during child labor. So the uterus smooth muscle is designed in such a way that as the fetus passes through the birth canal, it's going to increase the strength of those uterine contractions. Now, that is away from what set point is. Set point is going to be relaxed state of that smooth muscle. But for a desired result, which is to bring the fetus to the birth canal, we're going to strengthen those contractions. We're going to augment the strength of those contractions, and it's going to become greater and greater and greater and greater, moving us away and away and away from set point until birth has, been achieved, and then we can end that with the cessation of those contractions. So just two examples of what positive feedback is like. It's usually a little bit harder to conceptualize positive feedback, but I want you to think about it not really as the opposite of negative feedback, but as a part of negative feedback in some very specific cases. All right, we've got a third example of positive feedback here. This one involves the pituitary glands and the reproductive hormones. So the pituitary gland is considered one of our master endocrine glands, right? This is in the brain, and it's going to secrete a hormone called luteinizing hormone. So luteinizing hormone secreted from the pituitary has its effect on the ovaries, and what that does is to increase estrogen secretion from the ovaries. Now, this is typically a negative feedback cycle during most of a female's reproductive or our monthly cycle. This is usually going to result in a negative influence so that we can stop LH secretion, as we have enough estrogen secreting on the ovaries. But for a short period of time, this is especially important before ovulation, this cycle becomes a positive feedback cycle for a short period of time in order to encourage ovulation. So the estrogen release from the ovaries creates a feedback loop, a positive feedback loop that results in more luteinizing hormone from the pituitary, which causes more estrogen release from the ovaries. So this happens again and again and again until we have what's called a luteinizing hormone surge. This is called an LH surge, and that is responsible for ovulation. After ovulation occurs, this returns to being a negative feedback loop, which is what happens for the majority of the cycle. So only for that short period, it's usually about a day or so, this loop, otherwise a negative loop, becomes a positive loop to give us that desired result, and then it ends after that desired result. Okay, so we've talked about 3 specific examples so far: the blood clotting example, the uterine contraction example, and then here the pituitary gland and the female reproductive hormone example. So those are 3 of maybe 5 or 6 total examples of positive feedback that are found in the body, whereas negative feedback is much more common, many, many examples regarding most of our regulated variables. All right. Let's think about intrinsic and extrinsic regulation. And this is getting into what we described about the integrating centers and the loop of communication that allows for homeostasis. So there's a regulation of the processes within our various organ systems, and that can be brought about by 2 ways. Intrinsically means that cells inside the organ are responsible for some type of change. This is usually by way of local signaling factors. I'm not sure if you've heard of paracrine signaling, but it's basically when 2 cells are next to each other, they're neighbors, they can send signals to one another. That's kind of just local. It's not going to the entire body. It's not using the blood or the nervous system. It's just one cell to the neighbor. That is an example of intrinsic regulation of our various organs. Alternatively, we have extrinsic regulation, and this is where the brain gets involved by way of the endocrine system or the nervous system. So this is involving hormones, this is involving the blood, this is involving more widespread signals that are going to be going throughout the body, specifically engaging the brain. So this is kind of where the nervous system can tell the heart to increase heart rate. That's an example of an extrinsic regulation of the heart. However, intrinsically, whenever the chamber detects a stretch in blood, let's say we have a lot of blood coming back into one of the 4 chambers of the heart, the cells can detect that increase in volume, and they can increase their force or their ejection force. So there are 2 different ways that the heart, for example, can regulate itself: intrinsically, which is just local factors, extrinsically involving the endocrine system and the nervous system.